Hello. So today we are going to be doing Unit 4, Diffusion through a Membrane Pre-Lab. So diffusion through membrane is a two-part lab. So today we're going to be doing part 1. And this is one of the state-mandated labs that I've been talking to you guys about that is so important in this unit. And this is really the focal point of our unit, are the two sections of this lab. So today we're going to be doing the pre-lab for part 1. So let's go ahead and get started. So today's aim is how can we prepare for the New York State Mandated Diffusion Lab, because the fact that these are state mandated labs is part of what makes this so important. So these labs are required by New York State for you to finish the living environment class. They are covered on the Regents exam, and they are complex. So we spend a lot of time covering this topic. So let's go ahead and get started. In the diagram below, which letter indicates the part of the cell that carries out a function most similar to a function of the human excretory system? So we haven't talked about human body systems yet, but the excretory system is the system that includes your kidneys and your ureters, and it's what allows you to expel waste from your body via urine. So like the excretory system does in your body, removing waste, and allowing them to go out of your body, which organelle in the cell allows waste to exit the cell. B, that's that cell membrane. And we're talking all about the cell membrane because we're going to be building a model cell in this lab. And unfortunately, you guys are not going to be building a model cell yourselves because as I am recording this, they just announced that, we're going to be virtual for quite a while. And I did have a feeling that that was going to be coming up. But unfortunately, what that means is you guys will be watching a video of a model cell being built, because even I do not have the dialysis tubing to build this cell at home. So we'll be doing that all together. But for now, let's do this pre-lab. So which statement best describes the plasma membrane? And again, plasma membrane, cell membrane, same thing, of a living plant cell. It selectively regulates the passage of substances into and out of the cell. That's it. That's the answer. A. Uh, there are proteins and carbohydrates in the cell membrane, but as we know, it is a phospholipid bilayer, and that is critical to its selective permeability, the special property that selectively regulates the passage of substances into and out of the cell. It doesn't have the same permeability. That's the neat thing about it. It's selectively permeable. Certain substances are too large to enter and exit the cell freely, and we're going to examine that in this lab. And it has a double protein layer. No, it has a double lipid layer, phospholipid bilayer, and there are floating protein molecules. So it's the other way around. All right, so the answer to number 2 is A. Indicate that here for you. And the answer to number 1 is B, cell membrane. Okay, so dialysis tubing. I'm going to switch now to show you some notes. So, dialysis tubing, what is it? Dialysis tubing is a type of semi-permeable membrane. So dialysis, if you've ever heard of this process, is something that is used to mimic the human excretory system, the function of the kidneys. So dialysis is a procedure that is used to help people who have some sort of issue with their kidney functioning, to mimic the functioning of healthy kidneys. Their blood is taken out of their body and filtered externally through this process of dialysis. And this semi-permeable tubing is used in the separation of molecules from the blood during this process of dialysis, which is again functioning in the same way as the human kidneys are supposed to function. And we just saw in that do now question. Right, the cell membrane functions in the way most similar to the human excretory system. And this dialysis tubing is going to make our model cell. And the model cell is like made out of plastic. You'll see it, and that plastic is this dialysis tubing. But it's not just regular plastic, it is made out of a material that is similar to the cell membrane. So, which organelle is dialysis tubing most similar to? That's the cell membrane. How does it compare to this organelle? It is semi-permeable and it functions to separate molecules in the same way that the semi-permeable membrane allows only some molecules to pass through. How do molecules move across the cell membrane? Well, there we have it. Molecules must be small enough to cross the cell membrane, and they move from high to low, right? This is the process of diffusion. It always goes down the concentration gradient from an area of high concentration to an area of low concentration. And this process that allows molecules to move across the cell membrane is called diffusion. And it is the movement of molecules from high to low concentrations. And molecules can move across the cell membrane in other ways, but the process that we are going to be examining in this pre-lab and tomorrow's lab is diffusion. So this is an area of high concentration here. You can see, so this is the concentration gradient, right? Over here, it's really high, and then over here, there are no molecules. And that's what gradient means. It means high to low. So if you've ever seen like a gradient color scheme, right? It's sort of dark on one side and light on the other. That's what gradient means. And so you can see here, these molecules, they're all clustered over here and they're too close together. They're not comfortable. This is when you're getting right on the train car and everyone is right clustered by the door, and then you need to spread out, right? Fill in the empty seats, the empty doorways until you reach dynamic equilibrium, where all the molecules are spread out evenly. And this is how the molecules naturally move. This is what happens over time, right? The only thing that separates this and this is time. And this is the same process that when you put Kool-Aid powder into water and stir it up, or you put food dye into water and watch it change color, it is the process of diffusion that allows those molecules of dye or Kool-Aid to diffuse throughout the liquid water and become a homogeneous solution. So, and that's the definition of diffusion for your handout. And please draw this diagram at some point because this topic will unequivocally be covered on if there isn't a real Regents, whatever final there is that you will take in living environment, because this is one of the 4 New York State mandated labs. So there's only 4 of them. They're all really important. This is the first one. Part 1 of the first one. Okay, so here is our nutrient review. And you've got a word bank there: heated, polysaccharide, sugar, Benedict solution, starch, monosaccharide, glucose, and iodine. And this should be a review of our nutrient testing lab. So saccharide is another word for sugar. Starch is polysaccharide. Glucose is a monosaccharide. Since polysaccharides are long chains of monosaccharides, this makes starch a much longer molecule. So starch is a bigger molecule. Starch is a disaccharide. Specifically, but disaccharides are polysaccharides. They have multiple saccharides, but there are bigger sugar molecules than starch. But we are going to be examining starch here because we have an indicator for it in iodine. So iodine is an indicator for starch. In its beginning state, its color is amber. And if it's positive for starch, it will have a color change to black. And we are going to see that in today's pre-lab and in the lab. Benedict's solution is an indicator for glucose. In its beginning state, its color is blue. And if positive for glucose, it will have a color change to red, orange, yellow, green. In order to test for glucose, the solution must first be heated in a test tube in a hot water bath. And unfortunately, you guys didn't get to do that yourselves because it's pretty fun. So now we're going to take a look at a example, and let's go ahead and take a look. Okay, so we are going to watch a short little demo of this together, and I'm going to comment on it, and then we're going to do the last part of the handout together. So this is what tomorrow's lab is going to be. But in a class, what we would usually do before the lab is watch the teacher perform the lab in front of the class. So this is kind of what we're doing today. We're sort of pre-watching the lab before tomorrow we do the lab. You guys still don't get to actually do the lab. So it's sort of a world-class bummer, but let's make the best of what we've got.
Okay, so this is the diffusion through membrane lab for IS1. The first step is going to be adding the Lugol's iodine solution to the beaker. We will add 3 droppers full.
The diagram that you're looking at is illustrating this same thing. So we have iodine in the beaker. It's diffusing throughout. And you can see it first goes to the bottom and goes to the area of high concentration.
The next part of the procedure is to add the starch solution and the glucose solution to the Ziploc bag, which is our model for a membrane. So first we'll add the starch.
Interestingly, dialysis tubing and a Ziploc bag are both made out of a very thin plastic. And I believe this type of plastic works to function as a cell membrane because plastic is made from oil, which obviously is a fossil fuel. And fossil fuels are derived from really ancient, right? Fossil living creatures. So that is kind of morbid.
Solution. You can make your observations of that. And then we'll add the glucose solution. We'll be adding about the same amount of both to the bag.
Now,
Remember,
in our lab, we would add twice the amount of glucose. Although it looks like in this demonstration image, you do have the same amount of glucose and starch in the cell. So actually, he's doing the demo for your image. In the actual lab, it will be a little different.
the bag is serving as a membrane. It's not exactly the same as a cell membrane, but it still has essentially small openings in the bag that can allow for certain things to pass through based on their size. Each other, they make a dark purple color.
All right,
so here,
that was really quick. Okay, so he put the starch and the glucose in the model cell, okay? And he's allowing time to pass. So inside the bag is sugar and starch, okay? Both in a liquid form, okay? And at first, the outside of the bag has iodine, right? So in the beaker, there is iodine, which we just went over. Iodine is an indicator for starch, right? Lugol's solution is an indicator for starch. Outside the bag, a ton of iodine. Inside the bag, we have starch, and inside the bag, we also have glucose, okay? So now, at first, the bag is placed in the beaker, nothing happens. Then, a little time passes, and we have there a color change. So a chemical reaction has occurred. What happened? The iodine is small enough to pass through the semi-permeable membrane, to pass through the dialysis tubing. It diffuses into the bag and turns the starch inside a bluish black color. A chemical reaction has occurred. right? The iodine and the starch molecules have reacted with each other inside of the bag only. Inside of the bag only. And outside of the bag, we can see the iodine is still an amber color. So no chemical reaction has happened outside of the bag. The starch is too big, it seems, to diffuse through the semipermeable membrane. How can we tell that the starch is too big to diffuse through the semipermeable membrane? Well, if the starch had diffused through the semipermeable membrane, in the beaker, we would see this bluish black color. We would see a color change having occurred. So the iodine clearly got in the bag, but the starch couldn't get out. Let's continue to see the second part of this experiment. What happened to the glucose?
And so we can see that there has been a change to the color. Also do the glucose tests with the glucose testing strips. So just take one of these things out, show you the initial color. We'll dip it into the beaker solution and then let it rest for a second.
Okay, this is actually really cool because he's not using Benedict's solution, but what he has there, urine region strips, are the urine regent strips, excuse me, are what you would use to test a diabetic person's urine to see if their sugar is too high. Really interesting. Okay, so now the way we would test in the lab for glucose is by using Benedict's solution. But in this case, he's still using a glucose indicator. Why is he using a glucose indicator? Well, inside of the cell, we put starch, big molecule, polysaccharide, glucose, small molecule, monosaccharide. Outside the bag, outside in the back of, in the beaker, we put iodine. So we know the starch did not get out. How do we know this? The iodine outside of the bag stayed amber. However, the glucose is a smaller molecule. So did the glucose get out? Let's test.
And we'll look at the color. For the results of this, we'll actually take one that's been tested and compare it to one that hasn't been tested. So you have a your screen is the one that was put into the glucose solution. The one on the right is a clean test strip that has not been
So
put in.
similar to Benedict's solution, a blue is a negative, and you can see there that there was a color change, there was a chemical reaction, and so in the lab tomorrow, we'll watch the second part with, let's not watch a Star Wars trailer. In the lab video that we're going to watch tomorrow, we will see the full Benedict solution version, but let's go ahead and go over the answers to the final part of your handout. All right, so now we're going to diagram what we just saw. So in the experiment, in the beginning, we started with one model cell with two different things inside, with glucose and starch, and equal amounts of glucose and starch and iodine outside of the cell. So at the end, outside of the cell, we still had iodine, right? The solution was still amber. But what happened to the other molecules? So outside we still had iodine, but iodine also moved into the cell, right? We saw a color change happen. And inside of the cell, we still have our two starch. They definitely stayed inside of the cell, right? We didn't see a color change outside of the cell. And inside of the cell, we still have some glucose. We just, can assume that not all of the glucose got out, but as you may recall, there was a positive glucose indicator test that said that glucose got out of the cell. So here we can see the final state inside the cell. There is an equilibrium, right? So that is, we didn't test the percentage of iodine or glucose or starch in the solution. However, we can use our knowledge of biology and of chemistry here and know that molecules move until they reach a state of equilibrium. So that's what we're going to use to do the math here for our final state. So inside the cell, we're going to assume that we have 9 iodine. And there's also 9 iodine outside the cell, right? Half and half. Inside the cell, we have 4 starch. and there's no starch outside the cell. And we know this because the starch is too big to diffuse out of the cell, and we can support that with our observations, our findings in the lab. We saw that when the test was performed, the iodine outside of the model cell, the iodine in the beaker, did not experience a color change, did not react with any starch because there was no starch in the beaker. It was only inside of the plastic bag, right, inside of the model cell. However, the glucose inside the cell, there's still 4 glucose. Glucose moved from an area of high concentration inside the cell with plenty of glucose to an area of low concentration outside the cell where there was no glucose until it reached equilibrium for glucose. So we can see there it's half and half for iodine and glucose, but the starch didn't move in our final state. All right, let's go ahead. And if you were following along, you may have noticed I was using this during the video, so some of you may have already filled this in, but iodine is small enough to diffuse into cell. It diffuses and turns the starch inside a bluish black color. Right, a chemical reaction occurred. The starch is too big to diffuse through the selectively permeable membrane. It does not diffuse out into the surrounding solution. What will happen to the glucose? The glucose was small enough to fit through the selectively permeable membrane. This indicates that the glucose diffused, right? Because we know that the glucose is a monosaccharide. It diffused into the surrounding solution. It passed through immediately, which is why when you drink soda, it immediately diffuses through the membranes in your mouth, gets right directly into your bloodstream, and gives you that immediate sugar high. When you eat potatoes, it takes longer for your body to get the energy inside the potato to break those chemical bonds. When we test the outside solution for glucose and in the video we watched, they used a different indicator, but we used Benedict's solution. After heating it, right, the hot water bath is needed, the solution turns green. It shows a positive test. State one reason why a molecule may not be able to pass into or out of the cell. Too big. Positive or negative charge. But the one that we examined here was too big. All right, go ahead and complete your exit ticket and you are ready for tomorrow's part 1 of the Unit 4 diffusion through a membrane lab. All right, ladies and gentlemen, that's all I have for you today. Have a super fantastic day and I'll see you all tomorrow for the lab on Zoom. Bye, guys.